Great. All right. I just would like to make this video for the cell cycle control, and this is the diagram. And a lot of people ask me about this, so I would like to explain to you. So we have three cycles in this diagram: this one, and this one. And you will notice that this is the phases of the cell cycle, right? We have G1 here, S phase here, G2 here, and mitosis here. And then this cycle, the second cycle, it is for the G1 phase. All right, it is for the G1 phase, and this cycle, the outermost cycles, this one is for the mitosis phase. And by the way, you will also notice the two different arrows. Here is like a continuous arrow, right? There's no separate spaces. The arrow is like that, but this arrow is like separated, right? It's not continuous. So this discontinuous arrow means that these mitosis CTK, they are not walking, they only walk here. And the same goes for here. The CTK, they only walk during this time and they are inactive throughout this. So when you see this separated arrow, it means they are inactive, and when you see this type of continuous arrow, it means they are active. All right. Okay, we will start with this second cycle. This is for G1 phase, and like I said, G1 is before S phase. So what we do during G phase is we check if the cell has a good condition for the DNA to be synthesized or for DNA to replicate. So in the G1 phase, what we need is the CDK as well. We also need the cycling. So here, okay. So here, look at it carefully. We have G1 CDK here, and then G1 cycling here. So when this CDK and cycling combine, they become active, and we also use ATP, by the way. They become active, and here is a active G1 CDK cycling. It becomes active, and it works here. It works here, and then as soon as G1 is finished, cycling detach from the CDK, and it is degraded, and CDK is recycled. And then throughout this period, throughout S phase, G2 phase, and mitosis phase, this CDK is inactive, and only when it reaches G1 phase again, it binds with cyclin and it becomes active. Got it? Great. And now we move to the second cycle. So for the second cycle is for the mitosis phase. So you will see that this becomes mitosis CDK becomes active only when it reaches near the mitosis phase. Okay, so as soon as it reaches near the mitosis phase, so can you see this purple stick? This purple stick indicates the period where this CDK and cyclin are active. So only during this period, these CDK and cyclin will be added and will stimulate the mitosis phase. Do you remember that this cyclin and CDK together is called the mitosis promoting factor, and the mitosis promoting factor only promote the prophase and the metaphase. For the anaphase and the telophase, for the anaphase particularly, we need the anaphase promoting factor. So here it is in the middle of the mitosis phase. Okay, so this mitosis CDK, as soon as it reaches near the mitosis, it binds with the cyclin, it becomes active. We call this mitosis promoting factor, and it starts walking from here to up to here. This indicates the end of the metaphase. As soon as the metaphase is completed, we need anaphase promoting factor, and anaphase promoting factor continues the rest of the mitosis. Okay, this way. Great. And then here, look at it. While the anaphase promoting factor is stimulating the rest of the mitosis, the cyclin detached from the CDK. Now it's degraded, and CDK is recycled. It is recycled here. Recycle and go back to the mitosis phase. All right, so that is what you asked.